Hello, this is chapter 2 about how we see the invisible world, largely using our microscopes. So this first figure basically shows you two different kinds of ways that we can look at microbes. This first one here is a basic bright field microscopy image where the cells and whatever you're looking at, right, the cells are going to be stained darkly, and you're going to put that stain on the cells against a light background where the light is coming through the slide. And then we have a different kind here, which we can see it's a dark background and the bacteria are light, which is called a dark field image. Both of them are used for different purposes. We'll walk through some of those today. So here's a wavelength. So to understand light, right, we have different wavelengths of light, and the whole electromagnetic spectrum, visible light is just one piece. So let's just look at that first. So this is the whole electromagnetic spectrum. It's a form of energy that has different wavelengths. And in this region here, we have the wavelengths of light that our rods and cones pick up in our eyes. So we're able to see the wavelengths of energy here, but we're not able to see the wavelengths of the energy in all these other areas. So the electromagnetic spectrum contains cosmic radiation, gamma rays, X-rays, ultraviolet rays, infrared, radar, television, radio waves, AC circuits. So a lot of these things you've heard before, and there's just all this energy around us that we can't see. And you want to know from this that visible light is just one piece of a larger energy called the electromagnetic spectrum. And the different, what causes them to have different energies is basically their wavelength. All right, so let's look at the picture here. So every light, right, or the energy travels in waves. And one wavelength is the difference between basically just how far is one peak to another peak. The size of the wave is the amplitude. But really we're concerned with the wavelength. So if we have really far apart waves or kind of rolling like this, one wavelength is rather large. It has a low frequency and low energy. As the wavelengths come closer together, like this, for example, the waves travel faster, and that has a higher frequency. So what we want to remember is the amplitude is the height of a wave, and wavelength is a distance from one peak to the next. And the waves have different frequencies or rates of vibration. So if it's a larger wavelength and you have lots of space between, then that's a low frequency, low energy wave. And then if it's close together, we have smaller wavelengths, that's a higher energy. So if we look at the spectrum then, we can see that we have these in nanometers, right? Where that's our unit of measure. And we have our bluish form of light that our eyes pick up is around 400 nanometers. The red in this area is 700 nanometers. So which color has the highest energy associated with it, right? It would be the one with the smallest wavelength number, which would be our blue or purple light. And then ultraviolet would be beyond that. We can't see it, but it would be something that, animals can see, some insects can see ultraviolet, and that has a higher energy. So the reason why television and radio broadcasting is in the air all around us is that it's such a low energy wavelength that it doesn't penetrate into our bodies. It just, doesn't harm anything. So it's safe to be around. These higher wavelengths of energy get dangerous. So they have so much energy that they can penetrate into your body. So X-rays, for example, those go through your body. And they're not powerful enough to go through like bone, but they are powerful enough to go through your soft tissue, which is how we have X-rays. And X-rays are so in terms of clinically dangerous, right? Pregnant women are not supposed to get X-rays because that penetrating energy can actually harm the fetus or the embryo, because those waves can actually hit molecules and disrupt those molecules, right? So DNA being the one that we're very concerned about. Gamma rays and radiation, these are just higher energy. They're more dangerous for us to be around because the energy can penetrate into our cells. It can knock the DNA around. It can cause a lot of issues with mutations and cause cancer. So we want to make sure we avoid getting exposed to the short wavelengths. Longer wavelengths are no trouble. And then we can see visually, right? Our brain interprets these different wavelengths as different colors. All right, so another topic here is refraction. So a quality of light that comes in is that when it goes through a medium like air or water, it has one trajectory and one refractive index. When the light hits a different kind of material, it will bend because there's a different refractive index in a different material. And then you can see it picks back up with when it hits the air again. So this is a characteristic of light that it does refract and bend as it travels through different mediums. Another word we want to know is reflect, right? So the refracted light goes through the medium. It bends slightly away from its original trajectory. And then another word is reflected, where the light just bounces off that surface and just goes off that surface. So one can be refracted and then another, some of it can be reflected. This is just a cool picture of a pole looking bent, but it's actually straight as it goes into water because of the refractive index of air and water are different. Here's something on resolution and contrast. So resolution is the ability for your microscope or your eyes or whatever it is to be able to look at the fine detail, the ability to discern two objects that are very close together as separate. And so resolution is a way of thinking about clarity and how, resolved or clear it is. Contrast, though, it really shoots the light and dark into more depth. So contrast, you can see in these three pictures, contrast A has very little contrast. You cannot see really any detail or the background from the cell. Figure B has a more pronounced contrast where you can see a halo around the nuclei. We can definitely tell background from the cell. And this is a different form of contrast, basically just different ways that we can look at cells. There's phase contrast microscopy where you can really play with the contrast. So resolution is how clear it is, and then contrast is how the dark and light portions of the image could become outstanding. And I think that everybody that has a phone and takes pictures, plays with this stuff, understands probably what contrast is. Magnification versus resolution. Okay, we all know this, right? When you magnify something, it gets bigger. I love this picture of the little dog teeth. But we have the same magnification in both pictures, but the resolution is not the same. You have a more resolved image here than you have here. And so resolution is the ability to see fine detail or enable to discern 2 points that are very close together as separate. So clarity and detail. Whereas here you have a blurrier image because you don't have that separation of substances that are very close together. And so that's the difference between magnification and resolution. Back again to our microscope people, Leeuwenhoek in the first microscope. This little guy here, if you read comment B, even though Van Leeuwenhoek's microscopes were simple, they were powerful and better resolution than compound microscopes of this day. So he did a better job. So Robert Hooke was a man who used a compound microscope. So the microscope has changed and evolved a little bit, and he was able to see how living things all had individual units, and he called them cells. Cells really referring to the rooms of a monastery, basically dorm rooms that were called cells. Here's an image of looking for bacteria. So this is going to be very useful for you to realize that when you start looking for bacteria on a slide, under your scanning objective lens, it's going to not look very great. It's going to look like particles of sand, maybe just a little bit of background debris. But that's what you want to see when you're scanning. Then when you go to low power, you'll start to see a grainier image. 400, you start to get something that looks like this, but you really can't see the details of one cell. And then when you get to oil immersion, this is going to be, so this is high power. Oil immersion is the only way to look at bacteria in microbiology, because now you can sort of resolve those individual shapes, right? So if you look carefully, you'll see that these shapes are round little circles and not rods. And then the round circles, what's tricky is that some of them can be stuck together, like you can see a little bit of a 2 or 3 here together. So it's very important that you use the oil immersion lens to really look at your bacteria and decide like what shape they are and other features of it. So here's our resolving power. So resolving power depends on 2 factors, numerical aperture of the objective lens and the wavelength of light. What the numerical aperture is basically how much light gets into that tiny lens that's at the bottom of your objective lens. You can see that this picture here in A is going to be something like scanning or low power, because number one, I know because there's a very large working distance, okay? The least magnified objective, your lowest power, is going to have the biggest gap between your slide and the objective lens. So this is called the working distance. In number 2, as we go up in magnification, the objective lens comes closer to the slide, where working distance gets smaller, and the aperture, if you just look at this black line going across, it does decrease. So the amount of light coming into this glass region of the objective lens goes down. And then finally, when we get to like high power, you can see that our working distance is super tiny. Our aperture gets a lot smaller. And so this means that the resolving power gets less and less. So as less light comes into the objective lens, it's more unable to resolve. So what we want to do is we want to really put as much light into that objective lens as possible. The other resolving power issue or like factor is the wavelength of light, although we do not play with this. We don't actually we don't adjust these 2. This is just a fact of the microscope. So for our light microscopes, we have a resolving power of 0.2 micrometers. What that means is that when you look under the microscope, and let me just draw this out a little bit. Okay, so if 2 bacteria, right, I guess we have them here, if the space between them is larger than 0.2 micrometers, and remember this is like a length, right? 0.2 micrometers. If they're like 0.3 micrometers apart, then you can tell that they're 2 different cells. If they're like 0.1 micrometers or closer together, then what you're going to see is maybe something a little bit blurrier, a little bit like connected when they shouldn't be. Maybe you can see that they are separate cells, but it's not as good as a fully resolved image. And then unresolved is where they're so close together that you're just picking them up as one big blob. So that is going to be the limiting, basically our resolving power is going to limit, you know, how much we can resolve using our light microscope. So this is our light microscope. You're going to need to know this for forever for the exam and for lab. The ocular lenses are going to have a magnification of 10x separately. This is a binocular microscope, meaning that you have 2 ocular lenses to look for both eyes being open. Some microscopes only have 1, and that's a uninocular, but we have a binocular microscope. We have our revolving nose piece, objective lenses with different powers on them. I want to talk a little bit about the diaphragm and the condenser. So we know that light comes up through the illuminator, and before we hit the glass sample, we have a piece of glass called the condenser. Okay, so this is glass that's right underneath the stage, and what it does is it condenses the light. So remember, light sort of like refracts everywhere, right? Bends and goes off in different places. But this condenser tries to trap the light that's traveling here and focuses it so it comes up and focuses right through this hole in our stage to look at the specimen. So more light always equals more information. The diaphragm is going to be a piece of plastic that kind of is blocking, or it basically has a hole in it, right? So the diaphragm just looks like a piece of plastic, and it has an opening, and the light goes through this opening. And what you can do is you can control the opening size, kind of like the irises of your eye. So you can make it a really small opening, you can have it big. So that's going to control the amount of light going through to your sample. What this does is it really controls contrast, right? If you think about contrast, it really has to do with how much light is shining. Think about going outside on a really bright, snowy day. Oops, I was thinking contrast. And how everything is just blurred out because so much light is in there, hitting your eyes, that everything is washed out. That's what the condenser and diaphragm, if you have it completely open, sometimes that happens where your image is completely washed out. So you would want to like close it to make less light come in, and it really does help with the contrast. So I encourage you guys to find the knob. You can see it right here. Find the knob under your microscope, and you can play with that, and you can really see the difference with the contrast. So the job of immersion oil is to allow more light to enter into the oil immersion objective. So when we see with immersion oil, here's the microscope slide, which is glass. Here's the light source coming from glass. And what happens is if it hits the air, so where's the glass? The glass is going to be underneath the objective lens, right? There's a little piece of glass that allows the light to come in. This is glass, right? So we go glass, air, glass. So we don't want that air to interrupt the light, because some of that is going to refract away, right? We know that light, when it goes through different mediums, will bend. And so this immersion oil, what it does is it seals the two glass portions together, and it has the same refractive index as glass. So it bends light exactly the same as glass. So that way the light can just shoot right through the glass slide in a straight line, go through the immersion oil, and then continue on into the objective lens. And so that is what immersion oil is for. If you do not use immersion oil on your slides when you're looking at bacteria, you're going to lose a lot of information and it's going to be blurry. All right, moving on to dark field. So for dark field microscopy, they just simply put a block to block the light coming through. And so what it does is it makes a nice dark background, and it allows only the reflected or refracted light of the specimen to reach your eye. This is what a dark field microscope image looks like. And this is you'll see this again, but these sort of curly cells here, that is a microbe called Treponema pallidum. It's a bacteria. It is a spirochete. So it is one of those curly ones. And spirochetes are very tightly wound, and they're like a corkscrew. They move like a corkscrew. But these are most of the time stained with dark field microscopy, or use dark field microscopy to see. Here's some phase contrast microscopes just to see the difference. The resolution's the same. It's just the contrast of light is different. So we can really see different structures using that contrast, playing with contrast. All right, we get far greater detail. And our last kind of stain here is going to be an immunofluorescent stain. So this is more sophisticated and it requires more, like we're not going to do it in class. It requires a lot of sophisticated equipment. But what you can do, or you should understand the background. So what we have is an antigen. This can be anything you want to look at, you want to stain. So this could be a protein, it could be something on the bacteria. This example here is Neisseria gonorrhea. So this is the bacteria that causes gonorrhea. And what you do is you have a antibody. Okay, if you don't know what an antibody is, it's made by your immune system. It's very specific. This antibody binds to really specific proteins. And as a scientist, you would want to choose your antibody to attach to a certain kind of thing. So if we want our antibody, this black antibodies are Y shape. If you wanted to stick to something, right, you would purchase an antibody that recognizes Neisseria gonorrhea. And then on that antibody, the company makes a little fluorochrome that is attached to the antibody. So a fluorochrome is basically a fluorescent protein that will shine a specific wavelength of light when you activate it with UV. So this will actually give off a color when you shine UV light on it. And what you get is pretty cool. So you can get images like this, where in this image, fluorescent dyes have been absorbed by just a skin cell, an endothelial cell of bovine pulmonary artery. So this is a cow's little artery, and we can see that they put blue fluorochromes on the antibodies that recognized nucleic information, right, like DNA or RNA. Then they marked a green one, it's bound to actin, right? So you can see all the green areas is basically actin. And then you can see, do they talk about the red? Red is bound to and okay, sorry. Microtubules are marked by green and actin's marked by red. So the cytoskeleton of this is green and red. So you can see how you can really specifically see, where is my actin, where is my microtubules, where is my DNA, right? So you can locate things inside of a cell and highlight them with color. All right, I think our last microscope to talk about is something called an electron microscope, which is a very different machine. It is not something that you use your eyes to look at. You have an electron, basically a beam of electrons moving through this machine, coming down. Your sample would go in here, and it would basically, the electrons would hit your sample and be reflected right off, and these electrons would be picked up by a computer. You know, the equipment would pick up the pattern of the diffraction and reflection of the electrons, and then the image would come up on the screen. So you're not using your eyes at all. There are two kinds of electron microscopes: transmission and scanning. Transmission requires very thin sections to work with, and the advantage is that you can see inside the cell. So if you want to study the organelles, what kind of, where are they located, what's the pattern, how many are there, it creates a nice detailed two-dimensional image. Whereas a scanning electron microscope gives you really high detail of the surface only, right? You can only see outside, but these are really, really detailed, and it gives you a nice three-dimensional image of the object. The advantage of electron microscopes, like why would you use this, is because they're able to see things much, much smaller than what's seen with the normal microscope, like the ones with our eyes, right? Compound microscopes. If you remember back to the chapter 1, electron microscopes are able to see viruses, which are super, super tiny, and they're measured in nanometers. Remember that? So things that are measured in nanometers can be seen here. We can see sort of molecules and proteins with this, not yet our atom, right? I'm realizing I can't talk and write. See, anyway, you know, molecules. Anyhow, things that are measured in nanometers. Nanometers. All right, so for this, I just have some fun images to show you. So this is from your online reading. This is a biofilm showing different layers and different structures within that biofilm. And then this is just a color enhanced Staphylococcus aureus image. So the staffy refers to the clump-like arrangement. See, it's like a cluster of grapes. And then coccus refers to the fact that it's a round circle-shaped bacteria. Again, this is the genus Staphylococcus. The species is aureus. I just have some pretty pictures for you guys to look at. This is a head of an Antarctic mite. These are all three-dimensional, taken with a scanning electron microscope. And you can see it's been increased 1,500 times. So the most a light microscope that we use in class magnifies is 1,000, right? Using the oil. So this is 1,500. So you can see all the nice detail here. Here's a termite's head. Here is a spider. Here is an ant holding a microchip. I don't know how they did that. Here is a fly's foot. So you can colorize these things because it's just a computer image. These little guys here, probably plant pollen that have landed on the fly's foot. Here's a human lice. This is a lice knit. This is some adipose tissue, fat in our body. All right, so overview of the different kinds of light microscopes. We have our bright field microscope. You want to know you know, this is the one you're going to want to know the most of, the parts of the microscope, what they do, you know, the refractive index, blah, blah, blah. The dark field, just know what kind of images it makes, you know, just what's here is fine. Phase contrast, we're not going to do differential. Fluorescent, we don't do confocal, we don't do two photon. So just kind of know those. And then we have the two kinds of electron microscope images. This is going to, you know, again, the transmission, it passes through the specimen and we can get internal structures, where this one is for surfaces to look at 3D images.